Welcome to Study Loop.
This question asks, lactose digestion in E. coli begins with this hydrolysis by the enzyme beta-galactosidase. The gene encoding beta-galactosidase, LAC-Z, is part of a coordinately regulated operon containing other genes required for lactose utilization. Which of the following figures correctly depicts the interactions at the LAC operon when lactose is not being utilized? On the right, we see a figure depicting the LAC operon and the control elements involved, and we see that LAC-Z is going to be part of our LAC operon. And the question asks us what will happen when what will happen to the LAC operon when lactose is not present or not being utilized. So we have to look for the situation where lactose is going to be unavailable. And there are two situations, one where there is high glucose and one where there is low glucose. But in either case, the same thing will happen to the repressor protein, and that is specifically what we will be looking at for this question. So on the left-hand side, we see a figure with our question choices, and we are going to be investigating what happens to two of the mechanisms on the LAC operon. We will be looking at what happens to the repressor and what happens to the RNA polymerase. So we should know that the RNA polymerase, as depicted back in our first figure on the right-hand side, will only bind to the promoter region when our repressor protein is not bound to it. So we can only have one bound at a time. Either the repressor is bound to the operator or the RNA polymerase is bound to the promoter. Both can't happen at the same time. So if we look at the two situations here, we see that our repressor protein is going to be bound to the promoter region, or to the operator region. And this is because the repressor protein will only unbind in the presence of lactose. So our repressor plus lactose means that our repressor is going to be released only in this situation. So when the repressor is released, our RNA polymerase can bind. So given this sequence of events, we can look back at our answer choices to determine what happens when lactose is not there. So choice A shows us what happens in the presence of lactose, so it will not be the correct answer. And choice B shows us that our lactose is bound to the repressor, and we know that this is incorrect because if lactose is bound to the repressor, this should be doing what it is doing in choice A. So that is also incorrect. Choice C shows that the RNA polymerase is bound to the operator region, which we know will be incorrect based on the fact that we know that our repressor in this case should not have lactose bound to it, and thus should still stick to that operator region, meaning that RNA polymerase should not be able to bind. And finally, we have our choice D, which has the repressor but with an empty slot for lactose. So there's no lactose bound, which is correct if lactose is unavailable. So choice D is going to be the correct answer.